Welcome back, fellow mitochondriacs, for another episode of Cancer is a Mitochondrial Metabolic Disease. Today, we are going to dive into the next piece of the puzzle when talking about ivermectin. This is part two. In part one, if you remember, we talked about all of the different molecular mechanisms that were either downregulated or inhibited by ivermectin. And those signaling cascades are critical for setting up the Warburg effect and the enhanced glutaminolysis that is seen in cancer. But today, we're going to talk about how ivermectin directly affects the metabolism of both glucose and glutamine. So let's get into it. So as we talked about the effects on cancer stem cells and the pro-growth pathways. Today we're talking about glucose metabolism, glutamine metabolism, as well as how ivermectin is able to induce oxidative stress and apoptosis within cancer cells. So for the first paper I want to go over here, it was a paper that was published in February of 2023, and it's titled A Cellular and Molecular Biology-Based Update for Ivermectin Against COVID-19. Is it effective or not effective? And although this paper is about 2 years too late, 3 years too late for talking about its effect on COVID-19, I wanted to use the information that was found in this particular paper as a way to help us unravel some of the molecular mechanisms of how it could be effective for cancer. So we are currently in the middle of a giant glycolysis series, and we have been looking at all the steps of glycolysis, whether it be glucose uptake channels, whether it be hexokinase, whether it be phosphofructokinase, whether it be PFKFB. We're currently talking about pyruvate kinase. We're going to be talking about lactate dehydrogenase and subsequently these lactate channels. And what it says here in this paper, it says that ivermectin has been recently shown to play an important role throughout molecular networks by acting on key enzymes and energy metabolism pathways, particularly glycolysis phosphofructokinase platelet type, which is critical because we've seen that that is a very common isoform of PFK within cancer, isocitrate dehydrogenase, cytochrome B, NADH dehydrogenase 2, and ubiquinol cytochrome C reductase hinge protein oxidation in the Krebs cycle phosphorylation, and monocarboxylate transport 1, MC1, and MC4 in the lactate shuttle. So some of these are probably more critical to cancer biology than others. In particular, the blockade or the inhibition of PFK and the inhibition of these lactate transporters. If you remember back from 3PO lecture and talking about how they believe that that was an effector on this PFK initially, and then later they found that there was no effect on PFK, but really what was happening was it was actually blocking these lactate channels, and that was allowing for lactic acid to build up within the cancer cells, and then that would ultimately downregulate and inhibit all of these glycolytic enzymes, which ended up shutting down glycolysis. And so we see here that ivermectin is playing at least two major roles within the glycolysis and the kind of what would be in cancer a Warburg metabolism and shutting that down. Now, the next paper we're going to talk about is from April of 2022, and it says ivermectin accelerates autophagic death of glioma cells by inhibiting glycolysis through blocking GLUT4 mediated JAK-STAT signaling pathway activation. And it says here that ivermectin inhibited GLUT4 protein expression and overexpression of GLUT4, promoted glycolysis, and inhibited autophagic cell death in this U87 cells. Ivermectin inhibited glycolysis by blocking GLUT4, mediated the Janus kinase signal transducer, and activated the transcription JAK-STAT signaling pathway activation. Ivermectin inhibited tumor growth in vivo, decreased the protein expression of GLUT4, JAK2, hexokinase 2, and PFK1 in tumor tissues. So this paper, a subsequent paper, when talking about glioblastoma multiforme, they were finding that not only is it blocking PFK, but it's blocking glucose uptake channels, hexokinase, and these lactate transporters. And what I would say that the coolest paper that I was able to find when looking at ivermectin from a metabolic perspective, I'm going to move this over just slightly so you can see it better because my face is in the middle there. I want you guys to be able to see this better. We see here that this is an ovarian cancer model, and this paper was published in November of 2024, so fairly recently. Pharmacoproteomics reveals energy metabolism pathways as therapeutic targets of ivermectin in ovarian cancer towards 3P medical approaches. And it says here that molecular changes in tumor tissue of ivermectin-treated mice were established, and protein-protein interactions analysis showed that the main differential networks, including the TCA cycle, propionate metabolism, 2-oxocarboxylic acid metabolism, and other pathways, integrating our previous clinical ovarian cancer tissue and cell experiment data. This study found that ivermectin significantly interfered with the energy metabolic pathways of ovarian cancer, including glycolysis, TCA cycle, oxidative phosphorylation, and other related pathways. The significant efficacy of ivermectin will be applied to the treatment of ovarian cancer and personalized medicine. This has guiding significance for the clinical diagnosis, treatment, personalized medicine, and prognosis evaluation of ovarian cancer. And given that we know that the Warburg metabolism and enhanced glutaminolysis is seen as a hallmark of cancer, it likely is going to have far-reaching effects outside of ovarian cancer models. But we see here that in ovarian cancer, this is the untreated ovarian cancer, we see upregulations of glycolysis and TCA cycle. In particular, we're seeing upregulations of enzymes that are responsible for the conversion of alpha-ketoglutarate to succinate. And that's got very important ramifications. And the reason I say that is because if we remember looking at our kind of beautiful graphic from one of Seyfried's more recent papers, we see that glucose is going to be fermented into lactic acid. Okay. And that uses glycolysis and the Warburg effect to do that. However, glutamine is fermented to succinic acid. Okay. And you're going to see that ivermectin is going to shut that fermentation down to some degree. So if we were able to take what we found in all three of these papers, both this COVID-19 paper, this glioblastoma paper, this ovarian cancer paper, and we put it all together on a graphical representation, we see that ivermectin has inhibitory effects on the GLUT transporters, on hexokinase, on phosphofructokinase, on hypoxia factors, as we saw in the last video, on pyruvate kinase, as well as effects on glutamine fermentation. So let's take a look at some of the enzymes we see here that are affected by ivermectin. We see that there's a downregulation of IDH2, as well as SUCLA1, SUCLG1, and ODGH and DLST. Well, I had to do some searches because I'm not ultra familiar with all of these enzymes. And the SUCLG1 is a critical subunit of succinyl-CoA lyase. The SUCLA2 encodes for the beta subunit of this succinyl-CoA synthetase. And then DLST makes up part of the alpha-ketoglutarate dehydrogenase complex, which catalyzes the conversion of alpha-ketoglutarate to succinyl-CoA in the TCA cycle. Now, why am I taking the time to actually look at these gene products? Because that's what they found in this pharmacoproteomics study that are downregulated by ivermectin. Well, why do we care? Because those enzymes, this succinyl-CoA synthetase, for example, that's downregulated, is responsible for converting succinyl-CoA to succinate. And we know that ATP is made from that step. Succinyl-CoA to succinate, you see ADP is made into ATP. So that is the process Seyfried is talking about here, where ATP is made from the fermentation of glutamine to succinic acid or succinate. And that succinate is then transported outside the cell and contributes to the extracellular acidification. So if we were to look at this from our kind of classic glutamine graphic, if you remember back from the glutamine series, We looked at glutamine transporters. We looked at glutaminase. We looked at glutamine dehydrogenase. We looked at SLC7811. What we have not really talked about so far, except for a tiny little bit in the membendazole video, is that ivermectin will also block or downregulate this succinyl-CoA synthetase, which is going to inhibit the cancer cell's ability to make ATP and ferment into succinic acid. And that's going to be big for us. So I think that that is pretty amazing. Now, it goes further. As we talked about in that first slide, we're going to talk about its effects on glucose metabolism, which we did, its effects on glutamine metabolism, which we did, and we're going to talk about its ability to induce apoptosis through a mitochondrial mechanism. And I'm going to start with this paper here that was published in December of 2021. It says ivermectin induces apoptosis of esophageal squamous cell carcinoma via mitochondrial pathway. And it says here that our results determined that ivermectin significantly inhibited the proliferation of esophageal squamous cell carcinoma cells in vitro and in vivo. Furthermore, we found that ivermectin markedly mediated mitochondrial dysfunction and induced apoptosis of esophageal squamous cell carcinoma cells, which indicated that the anti-proliferative effect of ivermectin on this ESCC cells was implicated in mitochondrial apoptosis pathway. Mechanistically, ivermectin significantly triggered reactive oxygen species accumulation and inhibited the activation of NF-kappa B signaling pathway and increased the ratio of BAX and BCL2. Recent studies demonstrated that antiparasitic drugs can inhibit the growth of multiple cancers such as melanoma, ovarian cancer, breast cancer, glioblastoma. Here we found that treatment with ivermectin conspicuously suppressed the proliferation of these esophageal squamous cell carcinoma cells by MTT, colony formation, EDU incorporation assay, and were demonstrated in vivo of xenograft tumors. All control xenograft displayed a stronger Ki67 staining than the ivermectin-treated mice. Taken together, these data suggest that ivermectin inhibits the growth of esophageal cancer, both in vitro and in vivo. It has been reported that ivermectin inhibited breast and ovarian cancers by proliferation involving PAK1 ubiquination degradation and cytostatic autophagy by suppressing AKT, mTOR signaling pathways. We talked about that in the last video. The antiproliferative effect of ivermectin on glioma cells was implicated with ivermectin inducing cell cycle arrest and apoptosis. Here, our results on esophageal squamous cell carcinoma cells indicated that the antiproliferative effects of ivermectin on esophageal squamous cell carcinoma was significantly mediated via apoptosis. We found that treatment of ivermectin significantly increased nuclei fragmentation, apoptosis, and G1 cell cycle arrest in these esophageal cancer cells. Ultimately, what they found is we demonstrated that ivermectin inhibited the proliferation of these esophageal cancer cells by inducing mitochondrial dysfunction, suppressing NF-kappa B signaling, and promoted apoptosis. Our results suggest that ivermectin may be potential therapeutic target against esophageal cancer. And so just taking a look, graphically, what is actually happening here. And we've seen this in a variety of other medications used, and the way they do it is different. Some of these medications like DCA or melatonin or several other things will actually remove the blockade of energy flux through the broken mitochondria, increase reactive oxygen species. Ivermectin likely does something similar, or it could also, for example, as we saw here, it has blockades on complex 1 and complex 3 in the mitochondria. And when you block electron transport and proton transport through the electron transport chain, that allows this to be more leaky of reactive oxygen species and potentially cause reactive oxygen species that way. It seems like the mechanism is not obviously clear, but what happens is that mitochondrial dysfunction is further exacerbated. One of the reasons why cancer removes the mitochondria from the equation is because they don't want to allow this mitochondrial apoptosis mechanism to be able to be restored. And so a lot of these medications, including ivermectin, seems to restore that through the use of reactive oxygen species, and then ultimately cytochrome C release, and then the caspase system that's activated to activate apoptosis. That's further looked at when looking at this colorectal cancer model. This paper was published in August of 2021. It says ivermectin has a new application inhibiting colorectal cancer cell growth. It says the results demonstrated that ivermectin dose-dependently inhibited colorectal cancer SW480 and SW116 cell growth, followed by promoting cell apoptosis, increasing caspase 3 and 7 activity. Besides, ivermectin upregulated the expression of pro-apoptotic proteins BAX and cleave this PPAR and downregulated anti-apoptotic proteins BCL2 mechanism. Analysis showed that ivermectin promoted both total and mitochondrial ROS production in a dose-dependent manner, which could be eliminated by administering NAC or N-acetylcysteine to colorectal cancer cells. Following NAC treatment, the inhibition of cell growth arrest induced by ivermectin was reversed. Finally, ivermectin at low doses of 2.5 and 5 micromoles induced colorectal cancer cell arrest. Overall, ivermectin suppressed cell proliferation by promoting ROS-mediated mitochondrial apoptosis pathway and induced S-phase arrest of colorectal cancer cells, suggesting that ivermectin might be a new potential anti-cancer drug for human colorectal cancer and other cancers. And we see here, again, ivermectin is ultimately increasing ROS within mitochondria. It's upregulating these pro-apoptotic proteins, downregulating anti-apoptotic proteins. And then when that ROS damages the mitochondria terminally, it ejects cytochrome C, it ejects mitochondrial DNA, which is ultimately going to activate this caspase system and activate programmed cell death or apoptosis. And we see here this is apoptosis on these different cancer cells. And we see that in a dose-dependent fashion, we see an upregulation of apoptosis from 5% to 10% to 15% on this SW116. It seems it's even more pronounced. You see that at a 20 micromoles, you see a nearly 35% increase in apoptosis. And then you see that the reason that it's happening is because there's an increase in these caspase activity. Caspases are part of the programmed cell death pathways. We also see, and this is critical, this is exactly why I'm so anti-NAC for cancer patients, okay? Even if it lowers blood glucose, even if we believe that there's some good things it can do for disease prevention, etc., it's not good for cancer because it's what they use to control these experiments. So you see the control cell viability. We see that when we give ivermectin, it lowers the cell viability by 50%. And then what happens is if you give ivermectin plus NAC, you see that it actually is even a slightly higher growth than you would if you gave nothing at all, right? So not only does it reverse the effects of ivermectin, but it actually makes the cancer stronger. And this is what I call the internal shield system. NAC is a potent activator and enhancer of glutathione. And glutathione, although is an important antioxidant to help prevent diseases, when it comes to cancer, oxidative stress is part of the three-legged stool system, and it is something that we're actually trying to target. Moving on. In the discussion, it says, accumulating evidence suggests that ivermectin has anti-cancer activities against breast cancer, digestive system cancers, urinary system cancers, hematologic cancers, reproductive system cancers, brain glioma, respiratory system cancer, and melanoma. Considerable evidence indicates that both pro-apoptotic and anti-apoptotic BCL family proteins were implicated in apoptosis induced by ivermectin-induced apoptosis. This group, Song et al. In 2018 revealed that ivermectin induced glioma cell apoptosis by upregulating the expression of p53 and BAX, downregulating BCL2, activating cleaved caspase 3 and cleaved caspase 9. It further on says, in conclusion, we have demonstrated that ivermectin may regulate the expression of crucial molecules caspase 3, BAX, BCL2, PPAR, and cleaved PPAR in the apoptosis pathway by increasing reactive oxygen species, or ROS, production and inhibiting the cell cycle in the S phase to inhibit colorectal cancer cells. Furthermore, current results indicate that ivermectin might be a new potential anti-cancer drug for human colorectal cancers and other cancers. Exciting. And then the last slide I'm going to show here is a paper that was published in 2024. It says ivermectin inhibited bladder cancer cell growth and induces oxidative stress and DNA damage. It says here that our study showed that in vitro and in vivo, ivermectin inhibited the growth of bladder cancer cells. In addition, ivermectin could induce apoptosis, ROS production, DNA damage, and activate this ATM p53 pathway related proteins in bladder cancer cells. Conclusion. According to these findings, ivermectin may be a potential therapeutic candidate against bladder cancer due to its significant anti-cancer effects. And we see here that ivermectin is going to increase reactive oxygen species. It's going to ultimately activate these caspase-related proteins and apoptosis-related proteins. It's going to lead to DNA damage of these cancer cells, and it's going to lead to apoptosis or programmed cell death of the cancer cells. So pretty exciting that ivermectin has effects on glucose uptake and utilization. It has effects on glutamine utilization in the specific mechanism of glutamine fermentation from alpha ketoglutarate to succinic acid and succinate. And then it also, in addition to that, if that wasn't enough, it also induces oxidative stress and upregulates apoptotic proteins, downregulates anti-apoptotic proteins, and it ultimately leads to apoptosis of a variety of cancers if that wasn't enough. We have now covered a ton of things and important molecular mechanisms that show that ivermectin likely needs to be added to the standard of care sooner than later. In the next video, we're going to talk about how ivermectin synergizes with standard of care, how it synergizes with other metabolic inhibitors, and we're going to look at a couple of case reports looking at ivermectin used in clinical medicine. If you like videos like this, please like. If you have people in your life that could use information like this, please share it with them. And until next time.